Hello everyone, I am Vishwanath and today we are going to discuss a very important part of microbiology and medicine, that is poliomyelitis. And this disease is basically a disease of your muscles or your CNS and muscles, you can say musculoskeletal plus CNS manifestations are there on the basis of manifestation, right? So poliovirus is a virus of Picorna viridi. Okay, it is a virus of your family, Picorna viridi, right? And based on that, we can have three types of polioviruses, right? Type 1, 2, and 3. In some books, these names are not given. Just I'm telling you the names. If you want to remember, then it's well and good. But if not, then you can skip those names also. It is not given in your Apurva Shastri. But in some textbooks of medicine, it is given, right? So, type 1, this is called as Brunhilde mahogany strain, right? Brunhilde mahogany strain. Then we have type 2, that is Lansing and MEF1 strain. Type 2. The names are bit typical. Lansing and MEF1 strain, right? Lansing and MEF1 strain. And type 3 is Leon and Socket strain. Type 3, Leon and Socket strain, right? So these are the strains, the three types of polioviruses, right? And remember, this type 2 one, that is the Lansing and MEF1 strain, is the most antigenic one. Okay, this is the most antigenic, and maximum antigenic variations are also seen in this type of strain, right? Okay. So in polioviruses, we can have two types of antigens basically. One is the capsid antigen and one is the dense antigen, right? So we have in poliovirus, you can note two antigens. We can have C antigen, that is the capsid antigen, and D antigen, that is the dense antigen. And this D antigen is called as the type specific antigen. Type specific antigen, right? This is the type specific antigen. And if you talk about the incubation period of the disease, that is the onset of your the infection to the first appearance of the first symptom, right? This is the incubation period, and incubation period of this disease is 7 to 14 days, or you can remember 1 to 2 weeks, right? This is the incubation period. So I told about the types. I told about your antigens which are there, C and capsid and dense antigens, and your incubation period is 7 to 14 days. And the mode of transmission, I am writing MOT. Mode of transmission of the disease is a fecal-oral infection. This is a fecal-oral route. Fecal-oral route is the mode of transmission. It is the most common mode of transmission. Of course, you can have other modes of transmission, but they're very, very rare. Right? So when you talk about polio, it is a fecal-oral disease, right? The mode of transmission is fecal-oral mode, right? And this virus, basically, let's just a minute. Okay, so let's assume this is the virus. This is the V, that is the virus. So it is basically going to the gut, right? So it is going to the gut, and the multiplication of the virus, multiplication is occurring here. In the gut, the multiplication of the virus is occurring. Okay, and this virus uses CD 155. Okay, this virus uses CD 155. Let's assume this is the virus. Okay, this is the virus. I'm drawing it in. Yes. So it will have something like this CD 145, and due to the CD 145, it moves to the regional lymph nodes. It can attach and move to the regional lymph nodes, right? It can move to the regional lymph nodes, and via this regional lymph nodes, it will go to the blood. Via this regional lymph node, the virus will travel to the blood, right? And tend to the it is going the first time to the blood, right? It is going to for the first time to the blood. So this phenomenon in which the viruses first time appears in the blood is called as the primary viremia. So this is the case of primary viremia. So I told you it goes to the gut via fecal route, it enters the gut and it multiplies here via CD155. It moves to the regional lymph node, enters the regional lymph node. Via the regional lymph node, it moves to the blood, and this is the first time the viruses are entering the blood. So it is the primary viremia, right? And this virus is now circulating in blood. Let's assume this is the bloodstream. Let's assume this is the bloodstream, and you have this virus circulating in blood. So these viruses are circulating in the blood, and in the bloodstream, we know that we have some reticuloendothelial cells. Let's assume this the macrophages and all. Okay, these are the reticuloendothelial system. RES. I'm writing reticuloendothelial cells or phagocytic cells. This will basically eat the virus. This will basically eat the virus, right? And it via this, okay, it again goes to the blood, right? Of course, after eating, they'll move to the blood, right? So this is the second time. Second time they are entering the blood. So this is called the secondary viremia. So I told you about primary viremia. I told about secondary viremia. And the second times they enter the blood, they move to the brain and spinal cord. They move to the brain and spinal cord. So basically, this will be the place they will be going, the brain and the spinal cord, right? And along the nerve, okay, along the nerve in well, let's say your glossopharyngeal nerve will take, right? Along the nerve, it will move to the anterior horn cells of the spinal cord. Okay, it enters the anterior horn cells. You know about the anterior horn, posterior horn, if you have seen this representation of the spinal cord, okay, a butterfly type of thing. And anterior horn and posterior horn, you all know that, right? So it enters the anterior horn cells, and via this entry, it produces what is called as the flaccid paralysis. Flaccid paralysis. Okay. So paralysis is the complication in poliomyelitis. So it is basically causing flaccid paralysis, right? Spastic paralysis in which the body is more rigid. Okay, in flaccid paralysis, the body is more relaxed, right? So it's a flaccid paralysis disease, right? And this flaccid paralysis is occurring in the gray matter area of the spinal cord and brain. Okay, in spinal cord more specifically. So it is occurring in the gray matter area. So it is also known as gray matter myelitis. So this name is also important, also known as the gray matter myelitis. So I told you about the path taken by the polio virus to enter the blood, and via this primary viremia, and two times of it is entering the blood, primary viremia and secondary viremia is occurring. After that, it is entering the brain, the spinal cord, and the anterior horn cells, producing flaccid paralysis, which is called the gray matter myelitis. Right? Okay. Sometimes they ask you what is the earliest change noted. Earliest change occurring due to this infection of polio. Okay. What is the earliest change which is occurring? Of course, there will be many neurological changes. There will be degeneration of the neurons. But the earliest thing is the degeneration of Nizel's bodies. This is the earliest change. You know the Nizel's bodies, which are what are Nizel's bodies? If you have a neuron like this, it will have its nucleus like this, and the axon. These are the myelin sheaths. These are the nerve terminals or endings, and these are the dendrites. You all know the basic parts of a neuron. So in the cytoplasm, we have bodies like this, which are composed of rough endoplasmic reticulum plus ribosomes, and this form the nasal bodies. Okay, so what is the composition of nasal bodies? Rough endoplasmic reticulum plus ribosome. So this is the physiological thing. I'm not going to detail. So earliest change, you just remember, is the degeneration of the nasal bodies, right? So paralytic polio, basically we think that whenever there is a patient of poliomyelitis, we think that there will always be paralysis. It's not like that. The paralytic polio, you just remember that it is less than 1% only. Okay, less than 1% cases is only paralytic polio. Okay, and in paralytic polio, what type of paralysis I told you? Flaccid paralysis. You can say that it is a paralysis of the descending tract, not the ascending tract. Ascending tract is due to the sensory carrying the sensory signals, right? This is the paralysis of the descending tract. Okay, descending. It is not always bilateral. Okay, not always the one side descending tract is affected. Basically, and it's not that both sides will be affected. So it is the asymmetric one. So asymmetric, descending, asymmetric, acute, flaccid paralysis. So they ask you what type of paralysis is that. So I told you all the meanings of the term. Okay, descending, asymmetric, acute, flaccid paralysis. So this is the paralysis which is there, right? And if it involves the brainstem involvement causes respiratory depression. You all know that the respiratory centers are in the that medulla and the pons area and all. Okay, so involvement of this DRG, VRG and all. If that is involved, then there will be respiratory depression. Okay, respiratory depression, and there will be death, respiratory failure or respiratory depression and death, right? And in here, I told you earlier also, there is no sensory involvement. It's all motor. So I told you descending, right? No sensory involvement at all. You have to remember this point also. Okay, so this much of pathogenesis and clinical features are, I guess, sufficient for you for your exams. And after this, let's move on to the microbiological thing, and after that, we'll move to the vaccines. So we'll move on to the next part, that is the lab diagnosis of polio, right? How can diagnose it? Lab diagnosis. Lab diagnosis, right? So in lab diagnosis, you can say up to 1 week, up to 7 days, you can take a throat swab. Okay, and after when the patient will have 6 to 8 weeks of infection, right? So you can detect the virus in rectal swab or feces, right? And if the person is a immunocompromised person, you can detect the virus in urine. Okay, and this samples which we are taking, we have to freeze the samples, right? And the virus can be grown on primary monkey kidney cell line. That primary monkey kidney cell line, and we can observe the cytopathic effects like the Picornaviridae family. The cytopathic effect, that is the how the cells are behaving in the response to that virus, is shrinking of the cells, shrinking and crenation of the cells. Okay, and of course you can use the molecular methods like PCR. Okay, antibody detection, neutralization, multiplex PCR. You all know that this is a common thing. You have to write all this in all nearly all infections, right? Okay, and remember in polio virus, one important thing: humans are the only reservoir. There is no animal reservoir of this polio virus. Humans are the only reservoirs. Humans are the only reservoirs, right? It's not that animals are the reservoir. Humans are the only reservoir. I told you about lab diagnosis. I told you all the things orally. So I was talking about the sample collection. So a sample I talk that if it is 1 week, then you can take a buccal swab, or 6 to 8 weeks you can detect in your rectal, okay, swab, and also in feces. And in immunocompromised, I told you you can take urine, right? All of this I told you. And these samples have to be frozen, right? You can grow the virus on, I told you, primary, monkey, kidney cell line, and also you can do PCR, antibody detection, okay? And you can observe the cytopathic effect. Those who don't know what is cytopathic effect, it is the effect produced by the virus in the host cell. So whenever there is an infection by poliovirus, the cells will shrink and crenate. So the cells will be like this. Shrinkage and crenation of the cells is the characteristic cytopathic effect, not of only poliovirus, of Picorna viridae family, right? So this is lab diagnosis of the poliovirus. Of course, you can, if you are doing PCR, then you can also do multiplex PCR, right? And humans are the only, the reservoir of the virus, right? So these are the lab diagnosis part of poliovirus. And next we'll move to the vaccines for polio. Okay, this is an important part. We are talking about the vaccines for poliovirus. Vaccines, right? Vaccines for poliovirus. So we can basically have two types of vaccines. Okay, one is the injectable vaccine, one is the oral vaccine, right? So first we'll talk about the let's say here we're writing injectable vaccine, and here you're writing oral vaccine. This question is also important for your prof exams as well as your PG. Injectable vaccine and oral vaccine. So injectable vaccine is called the Salk vaccine. Just remember this name, Salk vaccine. And oral is called the Sabin. Like Salk and Sabin, right? So what is the regimen, dosing regimen of this vaccine? We give one vaccine at 6 weeks, 14 weeks, and 9 months, right? 6 week, 14 week, 9 months. Okay, this is the dosing regimen of injectable one. And oral one, we give 5 doses, right? So one is 6th week, 10 week, 14 week, 6, 10, 14. We give, then we give a booster. Okay, booster is given at 16 to 24 months. 16 to 24 months. This is the booster dose. And second booster is given at 5 years. Second booster, 5 years. So here you have the dosing regimen of the injectable one, and here you have the dosing regimen of the oral one. Okay, if this is clear, then we'll move forward. Then injectable vaccine, you can have 3 serotypes. The 1, 2, 3 are there. Okay, the 1, 2, 3 serotypes are there. I told you about the type 1, type 2, and type 3. Okay, and this is the dose format. And if you ask me about the dose, then 0.1 milligram dose is given intramuscularly. If you want, you can write 0.1 milligram. Sorry, 0.1. I'm not okay per dose. Okay, this is given intramuscularly. Okay, intramuscularly, and it is more safer. Injectable one. Injectable one is more safer, but it is a bit slow. Okay, it is a bit slow in action, and of course, as it is injectable format, we are giving parenterally. Of course, there will be complications, the general complications which you have while taking injection. But remember, this is expensive also. Okay, it is expensive also. It's not much like cheap. And short acting, slow and short acting. So these are the disadvantages, but one advantage is just safer, and also doesn't induce herd immunity. Doesn't induce herd immunity. Okay. Herd immunity means the individuals, there'll be protective titer of antibodies present in the individuals. Like in COVID times, we are talking about the herd immunity. Means if you achieve the herd immunity, then the infection can be controlled very easily, right? So this is called the herd immunity. So it doesn't induce a herd immunity. Okay, you give the vaccine to that person becomes immunized, but it is not like that. The community will be immunized, and of course, it is expensive. So we can't afford that in more. And if you want to know that how the vaccine is grown, then we grow in the same monkey kidney cell line. Okay, so this vaccine is also grown in monkey kidney cell line. This is also grown in monkey kidney cell line, right? So we talked about this vaccine, right? And about the oral vaccine, we're talking that this oral vaccine has a 6, 10, 14 weeks. Okay, this OPV, this oral vaccine is also called as OPV, oral polio vaccine. Injectable is called IPV. It's also given at birth. I forgot to tell you. Also given at birth, then 6, 10, 14 weeks. Booster is given at 16 to 24 months, and second booster at 5 years. Understood? Okay. There's some contraindications in which cases you won't give this oral polio vaccine, that is immunocompromised people, because the as you are injecting the virus, it can cause polio also in immunocompromised people. You know, the immunity is not that strong to contain that weak pathogen also. Then immunocompromised in pregnancy, old age. So in these cases, you will try to avoid this oral polio vaccine. But remember, the advantages are that it is a faster vaccine, the faster immune response. Okay, it will induce the faster immune response. It is more economical. It is cheap. Okay, दो बूंद you must be knowing that 2 drops of polio. Okay, दो बूंद जिंदगी के. Then it is long lasting. All the opposites of this. Okay, long lasting, and it will also induce the herd immunity. Induces herd immunity. So these are the things you have to know about the oral polio vaccine. Okay, I talked about this. And in India, you have something called as the Pulse Polio program. Okay, Pulse Polio program since some 1995 onwards, it is running 1995, 96 onwards. Okay, it is running in India, and in here, in less than 5 years of population, you are giving OPV. Okay, to all children less than 5 years. And some countries, if I talk about some epidemiology, then some countries are endemic for polio. Some countries are endemic for polio. You can remember stans, all the stans, like Pakistan and Afghanistan. So two countries are there, Pakistan and Afghanistan are endemic for this polio. And we have something in India known as the polio endgame strategy. Polio endgame strategy in India. Okay. So in this polio endgame strategy from 2019 to 23, it is continuing. We have three goals basically. Three goals. Okay. In polio endgame strategy, we have three goals. That is eradication, integration, certification, and containment. This is the third goal. So we have goals like eradication of polio. Okay, eradication. We have to eradicate polio. Integration. We have to bring together the communities for eradicating the polio. Your integration. Then we have to be certified and contain all the sporadic cases. Certification and containment. Okay, eradication, integration, certification, and containment. Okay, so these are the things. And okay, we have something called as just two are the special things which I will be telling you. One is VAPP and one is BDPP, BDPV. VAPP and VDPV. Okay, if you look at your book, then it is given. So it is a vaccine-associated paralytic poliomyelitis. Vaccine-associated paralytic poliomyelitis. The vaccine-associated paralytic poliomyelitis, and BDPV is vaccine-derived poliovirus. Vaccine-derived poliovirus. Okay, this thing. So VAPP is basically vaccine-associated poliomyelitis is due to your polio vaccine. You must be knowing that. Means you can understand from the names itself. Vaccine-associated poliomyelitis, not, paralytic poliomyelitis. So it is following OPV. So if the person is given OPV, then this condition can occur. Okay, and these are the strains. VAPP strains are there. VAPP strains are there. They are also OPV like OPV isolates, right? But this strains it can't circulate in community. These are not much dangerous. Can't circulate in community. Can't circulate in community, but this can cause your outbreaks and secondary outbreaks. This can cause sporadic outbreaks. Okay, so this is the thing. It's not much that dangerous. Like one case per 2.7 million would tell. One case can arise per 2.7 million doses, right? OPV. So if you give 2.7 million people OPV, then only one case will be noted, right? And this one case will be have a VAPP strain. As it can circulate, then we don't have to worry much also. Okay, and this is due to your oral polio vaccine. It is most commonly due to Sabin type 3 as compared to Sabin type 2. Okay, type 3 has a more potential to cause this VAPP as compared to type 2, right? Okay. Next, we have vaccine derived poliovirus. Okay, vaccine derived poliovirus. So, this what happens is that we are giving a live attenuated vaccine. Okay, we are giving a live attenuated vaccine. We are giving a live attenuated vaccine. So, this live attenuated vaccine is basically causing genetic changes. It is basically causing genetic changes and forming this vaccine-derived poliovirus. Okay, this live attenuated vaccine is basically forming the poliovirus by causing genetic changes. So we can have basically you have to remember the types. You don't have to remember go into much detail how is this forming and what is the pathogenesis, what are the exact things. But you have to just remember the types of BDPV. Okay, so we can have something called as CVDPV, that is the circulating one. C means circulating. Okay, CVDPV. Okay, it can circulate in community. It can circulate in community. Right, it can circulate in community and spread by fecal route. Unlike this VAPP, okay, it can circulate in community and spread by fecal route. Okay, and CVDPV is more dangerous than this VAPP. Okay, have to know this. And the majority are type 2. Majority are type 2 due to type 2. Okay, this was because of type 3 majority, but this is due to type 2, right? Okay, you just have to remember these things. Okay, you don't need to go into much detail or deeply understand these things. You have to remember these are just side things. You don't need to basically focus. We discussed the core things earlier. Okay, so no need to worry if you are not understanding. It's okay, right? And this CVDPV is there. Then IVDPV. Okay, IVDPV and AVDPV. Okay, IVDPV is the immunodeficiency associated. VDPV, immunodeficiency associated VDPV, and AVDPV is the ambiguous VDPV. Ambiguous. Just know the full forms, it's enough. Okay. So BAPV and VDPV is there, and these all the things I told you about the vaccines and about the pathogenesis, right? So we are done with poliovirus, and it's time for the fast forward revision. Okay, what I told you, we'll quickly see. Okay. So poliomyelitis, I was talking about poliomyelitis. It is a virus of your Picornaviridae family. Okay, you can have type 1, type 2, and type 3. So in type 1, we tell that it's the Brunhilde mahogany strain. Type 2 is called the Larsing and MEF1 strain, the most antigenic one. And type 3 is the Leon and Socket strain. I told you what is the strains. And two antigens are there, C antigen and D. C is called the capsid antigen, D is the dense antigen. D antigen is the type specific antigen, right? And incubation period is 7 to 14 days. I told you 1 to 2 weeks. Method of transmission is the oral, fecal-oral route, right? And the viruses enter the gut, and multiplication is occurring there. And via this CD155 marker, okay, in the cluster of differentiation 155, it is present on this, and it is moving and entering the regional lymph node. Regional lymph node is basically permitting those polioviruses which are expressing the CD155. So this viruses, you can tell a CD155 positive. You can use this in the diagnosis, even diagnosis of polio. You can use this in the diagnosis of polio. Even you can do an immunohistochemistry and this detect the CD155. But you have various other methods, so we don't use this generally. And regional lymph nodes, then it is entering the blood, it's the primary viremia, and entering the reticuloendothelial system, which is engulfed and again enter the blood. And again there is a secondary viremia. The secondary viremia, the virus is moving and going to the brain and spinal cord, where it's moving to the anterior horn cells of the spinal cord and affecting them. Destruction of neurons occur in the anterior horn cells. The first earliest change is the degeneration of Nielsen's body, I told you. It is causing the flaccid paralysis. Causing flaccid paralysis, I told you the type of paralysis is descending asymmetric acute flaccid paralysis. Descending asymmetric acute flaccid paralysis, which is also known as gray matter myelitis, right? And if it is involving the brainstem, if there is a brainstem involvement, then it is called respiratory depression. And there is no sensory involvement in poliovirus. I told you this. And if we talk about the lab diagnosis, you can take a sample. The 1 week infection, then you can take buccal swab. The virus will be detectable in the saliva, right? And if it is a 6 to 8 week infection, then rectal feces, which you have to frozen. This all samples basically have to freeze it, right? Immunocompromised people, you can take urine. And the virus is grown on primary monkey kidney cell line. Primary monkey kidney cell line. And if you want to take use molecular methods, then PCR and multiplex PCR are there. Antibody detection, you can use neutralization, complement fixation. Okay, you can just add over here that I didn't write. Neutralization test, complement fixation test, you can all use this. Okay. Then we can have your cytopathic effect. The full form is cytopathic effect. There's a shrinking and crenation of the cells, which is a common feature of your Picornaviridae, right? You can have vaccines for poliovirus. I talked about is injectable and oral vaccine. Injectable is called the Salk, oral is called the Sabin. It's called the IPV and OPV. Some FIPV is also available. That's the fractionated inactivated polio vaccine. Fractionated. Okay. So injectable vaccine can have 6 weeks, 14 weeks, 9 months. Okay. And oral vaccine you have to give one dose at birth plus 6, 10, 14 weeks. Booster at 16 to 24, and second booster at 5 years, right? Then grown in monkey kidney cell line. This injectable one is grown in monkey kidney cell line. Then 1 ml dose is given, 0.1 ml dose is given, IM, safer and slow. It's expensive, short acting, and doesn't induce herd immunity. But here, the oral vaccine is faster, cheap, long lasting, and induces the herd immunity. Pulse polio program, I told you about 1955, it was inaugurated, and here we are giving every child below the age of 5, we are giving oral polio vaccine, right? And endemic for polio, which two countries, Pakistan and Afghanistan. Polio endgame strategy we have in India, 2019 to 2023. Three goals are there: eradication, integration, certification, and containment. These are the three goals of polio endgame strategy, which can be asked. Then VAPP, vaccine associated paralytic poliomyelitis, and VDPV is vaccine derived polio virus. Okay, these things you have to remember. These two are separate things. BAPP is basically following OPV. You are giving a person OPV, you are giving a person OPV, and the polioviruses undergoes modification. You are giving after giving OPV, the poliovirus undergoes modification, forms BAPP strain, but it can't circulate in the community. Okay, circulation only happens of the BDPV strain, right? So BAPP strains are like OPV isolates, can't circulate in the community. One case is noted per this 2.7 million cases. Okay, so it is very rare, and Sabin type 3 is more implicated than Sabin type 2 in your BAPP. Okay, then we talked about BDPV, that is vaccine derived poliovirus. I told you about live attenuated vaccine is causing a genetic changes. When live attenuated vaccine causes genetic changes, it is forming the VDPV, that's vaccine derived poliovirus. Type of VDPV, you can see CVDPV, IVDPV, and AVDPV. So CVDPV is the circulating type, and it circulates in the community. Okay, circulates in the community and transmitted by fecal-oral route even. And more dangerous is this one, and then CVDPV is more dangerous than VAPP and majority type 2. IBDPV is immunodeficiency associated and ABDPV is ambiguous, right? So these are the things you have to remember as per poliomyelitis is concerned at this level. Okay, and this is a very important lecture. You have to understand this very carefully. If you want to connect with me, then you can use my Instagram ID. Okay, so this is my Instagram ID. You can just DM me if you have any doubts, and if you want to have any lectures, have any things, then you can just comment. Okay, and if you're liking the lecture, then do like and subscribe if you haven't. Okay, so let's meet in any other lecture. Bye and read well. Thank you.